Welcome to this course on genetics and public health. Genetics, the study of heredity and variation in organisms, has become one of the most powerful tools in understanding health and disease. From the way traits are passed from parents to offspring, to the molecular mechanisms of DNA, to the role of genes in chronic illnesses, genetics has revolutionized public health. Public health, on the other hand, focuses on preventing disease, prolonging life, and promoting health through organized efforts at the population level. When combined, genetics and public health explore how genetic factors influence population health, and how society can use this knowledge for disease prevention, early diagnosis, and effective treatment. This course is divided into two modules. Module 1, Foundations of Genetics. Module 2, Genetics in Public Health Practice. Let us begin with Module 1. Module 1, Foundations of Genetics. 1.1 Introduction to Genetics. Genetics is the branch of biology concerned with genes, heredity, and variation. It explains why children resemble their parents, why certain diseases run in families, and how populations adapt over time. At its core, genetics addresses three key questions. What is inherited? How is it inherited? How does it affect living organisms? 1.2 Structure and function of DNA. The genetic material of most organisms is DNA, deoxyribonucleic acid. DNA carries the instructions for building and maintaining life. Key features include: DNA is made of 4 bases: adenine, thymine, cytosine, and guanine. The sequence of these bases encodes information, much like letters forming words. Genes are specific sequences of DNA that code for proteins, which perform most of the body's functions. 1.3 Genes, chromosomes, and genomes. Genes are functional units of heredity. Chromosomes are thread-like structures of DNA and protein found in the nucleus. Humans have 23 pairs of chromosomes. The genome refers to the complete set of genetic material in an organism. The Human Genome Project, completed in 2003, mapped all human genes, opening a new era in biology and medicine. 1.4 Mendelian genetics. Gregor Mendel, often called the father of genetics, discovered the basic laws of inheritance. Law of segregation: each individual has 2 alleles for a trait, and these separate during gamete formation. Law of independent assortment: genes for different traits assort independently of one another. Law of dominance: some alleles mask the expression of others. These principles explain inheritance patterns of many traits and diseases, such as sickle cell anemia and cystic fibrosis. 1.5 Beyond Mendelian genetics. Not all traits follow Mendel's simple rules. Examples include: polygenic inheritance, where multiple genes influence a trait, such as height or blood pressure. Multifactorial inheritance, where genes interact with environment, as seen in diabetes and heart disease. Epigenetics, where changes in gene expression occur without altering the DNA sequence, often influenced by lifestyle and environment. 1.6 Genetic variation in populations. No two humans are genetically identical, except identical twins. Genetic variation arises through mutations, permanent changes in DNA sequence. Recombination, during sexual reproduction. Migration and genetic drift, affecting gene frequencies in populations. Variation is essential for evolution but can also lead to inherited diseases. 1.7 Genetic testing and counseling. Genetic testing examines DNA to detect mutations or predict disease risks. It is used in diagnosing inherited disorders, identifying carriers of genetic diseases, predicting risks for conditions like cancer, guiding personalized medicine. Genetic counseling helps individuals and families understand test results and make informed health decisions. In Module 1, we explored the foundations of genetics, including DNA, genes, inheritance, Mendelian and non-Mendelian principles, genetic variation, and testing. These form the scientific base for applying genetics to real-world health challenges.